Okay, this is the Professor, CBMD PhD, and today's Friday, November 24th, 2023. It is 5:31 and 42 seconds, AM Eastern Standard Time, and today's research, cell division and cancer. So let's hop to it here. The division of cells and cell cultures and cancer, date November 17, 2023. Source, ETH, Zurich. If cells and cell cultures grow while being treated with division-suppressing agents, their growth becomes excessive and they permanently lose their ability to divide. However, if the cells are treated with a combination of division inhibitors and growth inhibitors, they remain capable of dividing after these substances have been discontinued. The findings can be transferred to certain cancer therapies, but first need to be clinically tested and confirmed. In multicellular organisms, such as humans, the growth of cells must also be coordinated with their environment so that cells are present in the right number and size to form functional tissue or organs. Cell growth is therefore strictly regulated and takes place only when certain growth signals are present. As cancer cells are different, they grow unchecked, they divide over and over again, and they don't react to stop signals from their environment. Studies show that uncontrolled growth is not only an advantage for cancer cells, but also a weakness. The scientific investigators and researchers show how cell growth influences cell function. They are also investigating what happens when cells exceed their normal size and enter a state that the researchers refer to as senescence. In this state, the cells are preternaturally large and lose their ability to divide. Nevertheless, they are still active and can influence their environment, such as by releasing messenger substances. As senescent cells are found in normal tissue and play an important role in the aging process. However, senescence can also be induced with chemical substances, and because it leads to a loss of the capacity to divide, it is a goal of certain cancer treatments. Researchers investigated whether excessive cell affects cellular functions in senescent cells. They treated cancerous cell line and breast cancer cell line with substances that inhibit growth and division. When they used only division-suppressing substances in their cell cultures, the cells were indeed no longer able to divide, but they continued to grow and went into senescence. As a result, they permanently lost their ability to divide. This effect persisted even after they discontinued the division inhibitors. An important reason for the loss of the ability to divide is that the enlarged cells no longer repair damage to their genetic material, such as double-stranded DNA breaks. Such breaks always occur spontaneously when a cell duplicates its genetic material prior to cell division. In addition, these cells cannot correctly activate a key signaling pathway, p53, p21, which is critical for a coordinated response to DNA breaks. As a result, the damage is not repaired efficiently enough. What this means for enlarged cells is that numerous irreparable DNA breaks accumulate during division, to the point where division is no longer possible. When the researchers treated the cells with division inhibiting and growth inhibiting substances simultaneously, the cells were able to divide and multiply normally again after both substances were discontinued. In cancer therapy, this is precisely what you don't want. As growth and division inhibiting agents are already being used in cancer treatment, based on the observations in cell cultures, they would expect an increased relapse rate when treating a tumor with division inhibitors and growth inhibitors at the same time. It would make more sense to first use a division inhibitor than a drug that further damages the DNA of the cells and makes division completely impossible. The researchers have tested their new findings only on cell cultures with both growth and division strongly dependent on the cell environment. The team cannot transfer these results directly to a clinical setting. The trials with organoids or on tissue samples are thus needed, first to better test the potential treatment. Clinical studies investigating various combinations of division inhibitors and other medications are also underway. As studies show that cancer cells with hyperactive growth are sensitive to treatment with division inhibitors, as these substances are already being used to treat certain types of breast cancer. The new findings could have a long-term impact on cancer treatment. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Nature of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.